happen.
We've all lived through the most catastrophic health crisis since decades, the COVID-19 pandemic. It was a time of fear, uncertainty, mayhem, wreaking havoc on economic and healthcare systems on a global scale. By the end of 2020, COVID-19 had killed more people in 1 year than any infectious disease had done for over a century. The journey of developing, testing, manufacturing, and distributing safe, effective vaccines within a single year is unheard of in modern science and possibly one of humankind's greatest accomplishments. Even before the pandemic, vaccines were saving 2 to 3 million lives a year. But how do they work? To understand this, we need to know how our body's immune system functions. There are 2 ways to acquire immunity: through natural infection and through vaccination. Our immune system has 2 lines of defense. When a pathogen tries to enter our body, our innate system that stands guard at the gateways to our body quickly blocks the foreign invader. It doesn't need to have seen the virus before to protect us against it. However, smart or powerful pathogens can surpass this. This triggers the adaptive immune system, which is strong and specific. T cells work to recognize specific pathogens, while B cells produce virus-specific antibodies, which are proteins that neutralize the pathogen by binding to the outside of a virus. However, the adaptive immune system can be slow, giving time for the virus to infect the body's cells, replicate, and make us sick. This is where vaccines come in.
Vaccines stimulate the immune system to develop long-lasting immunity against specific pathogens. The goal of a vaccine is to elicit an immune response against a weakened virus, a killed one, or specific proteins of the virus. Though the pathogen is harmless, the adaptive immune system still responds to it with B and T cells. If the individual is exposed to the virus again, the adaptive immune system retains memories of this virus, and a much quicker, stronger secondary immune response will occur. The pathogen will be defeated before it can cause damage.
Viruses cannot survive on their own. They replicate or make copies of themselves within cells of living hosts.
Just like how a puzzle piece fits perfectly with another, the surface protein on a virus must latch onto a particular receptor on the surface of a cell in order to infect it. This is also why antibodies specific to the virus bind to it, so it cannot latch onto other cells. For the COVID-19 virus, the infamous spike protein on the surface of the virus allows it to attach to host cells. The tip of the spike binds to receptors on the host cell's surface.
There are some traditional vaccine technologies that scientists have used to fight disease for centuries. In some vaccines, there's a whole live disease-causing virus that has been weakened under laboratory conditions. This is called a live attenuated vaccine. Because the virus is still alive, it can stimulate an excellent immune response that is almost as good as a natural infection. However, there are safety and stability concerns. There is potential for the virus to reawaken, which is dangerous for immunocompromised peoples. If the virus is not just weakened, but killed altogether, it is called an inactivated vaccine. This has no risk of infecting people or causing disease, but it can still induce a good immune response. The Covaxin COVID-19 vaccine is an inactivated vaccine. The Novavax COVID-19 vaccine is a protein subunit vaccine. It presents a specific, synthetically produced protein of the virus to the immune system without viral particles. It may include an adjuvant that enhances the immune response, like how adding flavoring to a dessert makes it tastier. During the pandemic, there was actually something amazing happening: the development of new and revolutionary platform technology. Instead of making new vaccines for every virus, platform technology can be used as a template to create multiple vaccines, needing only the genetic sequence. The manufacturing, storage, and dosage can be applied to all vaccines of the same platform. This reduces development time and cost. The COVID-19 pandemic was the first time the world saw mRNA vaccines being brought in the market. This includes Pfizer and Moderna. So how do they work? We all know that our genes are made of DNA.
This is basically the raw code that defines us, our physical characteristics, and the proteins that our bodies produce for various purposes. When the process of creating proteins begins, DNA first has to be converted into mRNA or messenger RNA that can then be converted into proteins. It's like having an encrypted message, converting it into a form that can be understood, and then carrying out what needs to be done.
mRNA works by directly giving the body cells instructions to create the spike protein. This is displayed on the surface of the cell, and immune cells recognize the foreign pathogen, provoking an immune response. mRNA is unstable, so it has to be encased in fatty acid compounds or droplets while it is injected into our body. One misconception is that mRNA changes our DNA, but this is false. mRNA does not enter the nucleus of our cells. It can carry out its function in the cytoplasm. Another way to get genetic information into the body is by using a virus itself. An adenovirus is a type of virus that gives us a cold.
In a vaccine, however, it is modified. The gene that allows it to replicate or make copies of itself is removed, and it is replaced by a gene that tells cells to make a particular protein, the spike protein.
A viral vector acts as a vehicle, or simply the packaging, to give instructions to our body cells to create the spike protein. The immune system again finds the protein it doesn't recognize and reacts against it. The Oxford AstraZeneca and Johnson & Johnson vaccines are viral vector vaccines. To test for vaccine safety and efficacy, clinical trials were conducted. Each phase has a different purpose, and only after completing one phase can the next begin. So how was the COVID-19 vaccine development so rapid? To speed up their work, scientists had a brilliant idea. What if the various steps to develop a vaccine could be done simultaneously instead of one after the other? The research, trials, manufacturing, and distribution of COVID-19 vaccines could overlap instead of being done one after the other. It is important to understand that every single vaccine and vaccine technology played a critical role to combat this virus and to save lives during the pandemic.
This is a story of a race, not against different vaccines or their manufacturers, but of everyone together, racing against a virus that devastated the world. Most of us are already vaccinated against COVID, but another contagious respiratory virus we should be aware of is influenza. You may know this as the flu. Like COVID-19, influenza virus strains change and evolve rapidly, causing deaths, hospitalizations, and disease transmission, especially in target groups. Two types of the influenza virus, A and B, circulate around the world every year with new dangerous strains. Seasonal influenza vaccines are regularly reformulated every season to target these strains. This is why it is important for you to get vaccinated once a year. Vaccines are safe and effective, the best tool to prevent influenza illness and transmission. Protect yourself and others.